Hi everyone, my name is Monjo Claire, and today, alongside with the rest of Group 5, we're going to be presenting to you hepatitis C. So let's get right into it. I'm going to be going over the background, things like what is hepatitis C, how it was discovered, some of the pioneers that were integral to the discovery of HCV, and what is the global health burden that we've faced and are currently facing today. So, what is hepatitis C? Well, hepatitis C is just a liver disease that is caused by the viral infection of the hepatocytes, which are the main type of cell found in the liver by the hepatitis C virus. Hepatitis C belongs to the family Flaviviridae and the genus Hep C virus. Other Flaviviridae viruses examples include yellow fever virus, West Nile virus, and dengue virus, all of which we have learned in class and all of which we still study extensively because these are pathogenic to the human race. Hepatitis C is a group 4 virus. It's positive sense single-stranded RNA that once it enters the host cell can start translation right away. Hepatitis C is also an envelope virus that is made up of the host membrane that it steals from the host cells once it infects them and leaves, and these E1 and E2 glycoproteins, which play important roles in carrying out viral processes that we're going to go over later. There are currently 7 hepatitis C genotypes and 84 hepatitis C subtypes recognized by the CDC. And hepatitis C can only infect humans and chimpanzees naturally, meaning it has a very specific host range. Um, so how was hepatitis C actually discovered? Well, jumping back to 1975, Dr. Harvey J. Had discovered this novel type of hepatitis that was found in patients who had just received a blood transfusion. Once he had proved that this virus was not hepatitis A or B, but it was a type of hepatitis, he named it non-A, non-B hepatitis. He then collected plasma and serum samples from these infected patients, and he injected that plasma and serum into test chimpanzees. And the results were that every single chimpanzee had developed chronic hepatitis, and that virus actually flourished in their bodies. Um, 14 years later, Dr. Michael Houghton and his colleagues actually cloned a small fragment of this non-A, non-B hepatitis genome from the infected chimps, and they used it to run diagnostic tests on human blood. After these tests, they had decided to name this new virus, hepatitis C, which is where the name comes from. Dr. Charles Rice is also credited for pioneering novel methods of studying hepatitis C with liver humanized mice. So these mice had humanized livers, and he used those mice to study hepatitis C and make advances. His research awarded him the Lasker Award 2016 and the Nobel Prize in 2020. One interesting fact is our very own professor, Professor Melody Lee, did her postdoc under Dr. Charles Rice at Rockefeller. So what is the impact of hepatitis C on global health? Well, it is estimated there are 58 million people in the world with chronic hepatitis, and that's almost 1% of our world population. And with 1.5 million new cases a year, this virus is on the rise. In 2019 alone, it is estimated that there were 300,000 people killed because of hepatitis C. So this virus is has a very high mortality rate, and that's one reason we continue to study it. Another reason that this virus is very dangerous is that 75% of the people that are infected with hepatitis C don't even know that they're infected. So we need to get tested, and that should help us. But eradication of this virus is very, very unlikely without a vaccine. And currently, to this day, there is no vaccine. So I'm going to pass it over to my group mates who are going to go more into depth on this virus. Thank you.
Hello, my name is Austin, and I'll be going over major outbreaks, followed later by the HCV life cycle. Here I highlight the major outbreaks in the United States from 2008 to 2019. Now, within this time, there have been 43 outbreaks and 328 outbreak-associated cases. Now, these outbreaks occurred primarily in healthcare facilities, such as hemodialysis settings and long-term care facilities. Now, this is most likely due to irresponsible and unsanitary practices.
Hi, my name is Jacqueline, and I will be taking us into the overall research question for our presentation. Now that we understand the global impact of the disease itself, we want to know why hepatitis C is still prevalent today. And in order to answer this question, we have to look more into the structure and life cycle of the virus, its strategies of viral antagonism and evasion of host immunity, and its etiology and transmission, so that we can understand why there are still limitations to treatment and still no vaccines. First, looking at the structure of the virus, it is an enveloped positive sense single-stranded RNA virus that is spherical in shape and about 40 to 80 nanometer in diameter. There is a glycoprotein envelope made up of E1 and E2 heterodimers and a lipid membrane. Underneath is the capsid shell that surrounds the nucleocapsid-protected RNA genome. HCV infects hepatocytes, which make up 80% of liver mass. And this is relevant because HCV also exists as a hybrid lipoviral particle that resembles very low density lipoproteins, or VLDL. VLDLs are produced normally by hepatocytes and they circulate in our bloodstream. For HCV, the lipoproteins are taken from the host hepatocytes as HCV hijacks VLDL secretory pathways as they exit host cells. Later, you will be able to see how the interactions between HCV and host lipid metabolism affects HCV pathogenesis.
All right, so now I'll be going over the life cycle of hepatitis C. Now, the process of hepatitis C entering a hepatocyte is extremely complex and not fully understood, but the first step involves a lipoprotein, which alters its shape to expose the E1 and E2 glycoproteins of the HCV virion. Now, there are four main glycoproteins that this particle attaches to, and these are the SRB1 receptor, the CD8-1 receptor, and the CLDN1 and the OCLN receptors. Now, these receptors form the clathrin-coated pits. Now, these other receptors serve to aid in attachment to these receptors in the virion. Now, once the clathrin-coated pit is formed, the virus is able to enter through the hepatocyte through the clathrin-mediated endosomal pathway, as shown here. Now, as the positive single-stranded RNA enters the cytoplasm, it then navigates the ribosomes on the rough endoplasmic reticulum, where it engages in protein synthesis. Now, the result of this synthesis creates one long polyprotein, which is composed of both structural proteins and non-structural proteins. Now, this polyprotein is cleaved by viral proteases such as NS2 and NS3, which were brought in by the host virion upon entry into the hepatocyte. Now, replication of HCV starts with the formation of a replication complex, which involves both viral and host proteins, and this protects the virus replication from other environmental factors. Now, NS5A and NS5B are two viral proteins that serve as RNA polymerases to form the negative single-stranded RNA, which acts as a template for the positive single-stranded RNA. Now, there are other viral proteins that are important for this process as well, as shown in this legend down here. And this cytosolic lipid droplet is very essential as well, as it acts as an anchor for replication and helps in forming the lipoprotein particle for the HCV virus. Now, interestingly, HCV has two types of variants formed by this complex, the low-density lipoprotein variants and the high-density lipoprotein variants. Now, this low-density variant is associated with infection, while the high-density version is not. However, the high-density version is associated with chronic hepatitis C infection and can possibly damage the liver. Finally, the HCV envelope is derived from host cell membranes, specifically the ER and the Golgi apparatus. The E1 and E2 glycoproteins are then inserted into the ER membrane and transported to the Golgi for further modification by glycolization. The virions are not infectious at this stage and require further maturation, which involves cleavage of the viral envelope proteins by host cell proteases. Once the virion is fully mature, they are released into the extracellular space to infect new hepatocytes and continue the HCV life cycle.
Moving on to how viruses evade detection by host cells, we first have to talk about the innate immune response to HCV. There are different pattern recognition receptors that recognize the viral genome. RIG-I recognizes uncapped single-stranded RNA, TLR3 and MDA5 recognize the double-stranded RNA intermediate. Both of these pathways lead to the expression of type 1 interferons, which then signals to neighboring cells to start expression of ISGs. There's also PKR, which is another pattern recognition receptor that recognizes double-stranded RNA, and that inhibits translational shutoff. One of the ways that HCV can evade host immunity is by antagonizing certain proteins. For example, Viral proteases NS3 and 4A can cleave host proteins that are important to the first arm of interferon response. This evasion of innate immunity means that the virus can successfully stop cell death and remain in the host for a longer period of time, possibly leading to a more persistent infection or a chronic infection. We'll talk more about chronic infections a little bit later. HCV can also evade detection by adaptive immunity by hiding conserved antigenic epitopes under the E1-E2 glycoprotein envelope. E2 has a hypervariable region that can undergo rapid mutation, and because of this rapid mutation, it can act as a decoy for the adaptive immune cells and antibodies, allowing HCV to escape detection.
Hi, my name is Yusuf Mathkour, and today I'll be talking about the etiology of hepatitis C. First, I'll be discussing the transmission of the disease, before then talking about the effect on host immunity and how this leads to either acute or chronic infection, before finally talking about the effect of chronic infection on host lipid metabolism and how this leads to steatosis and other liver diseases. Transmission of Hep C. Hep C can be transmitted in a variety of ways. The most common way of transmission is associated with using unsterilized needles. As you can see by the image on the bottom left, Hep C is very prevalent, with some sources estimating 71 million infected individuals, mostly being in the Eastern Mediterranean region, the Western Pacific region, and the European region. The image on the bottom right shows an estimated total new HCV infections starting from 1982. You can see it gradually rising before taking a massive drop in 1989 due to doctors understanding the importance of screening blood for HCV before transfusions. However, recently it's slowly beginning to rise again. Acute Hep C infection is associated with the first 6 months of infection and is screened through a blood test. In about 1/4 of the cases, Hep C is destroyed by your body's immune system, and symptoms include fever, dark yellow urine, and so on. The graph on the bottom left shows as HCV RNA enters your body, your ISGs get turned on, leading to an eventual T-cell response. ALT is an enzyme that is produced with liver damage, so you can see how when HCV RNA is high, there are high levels of ALT, but when HCV RNA is low, there are low levels of ALT. Chronic Hep C infection occurs after 6 months of infection and is screened through a blood test. Doctors recommend immediate treatment or there's an increased risk of liver failure. Treatment includes using DAAs, which will be discussed later, and is effective around 90% of the time. Chronic Hep C can lead to future complications such as cirrhosis, liver failure, and liver cancer. The bottom right graph shows during a chronic HCV infection, the T-cell response is weak, which will be discussed in the next slide, and ALT is constantly being produced, showing an increased risk of liver failure and HCV RNA staying in your body. Your adaptive immune response during an acute HCV infection plays an important role. In an acute and cured response, CD4 T cells call upon CD8 T cells. CD8 T cells stay for decades in case of second exposure. As you can see in the graph on the top right, CD4 T cells call upon CD8 T cells, leading to minimal virus load and T cells that are ready for a second response. However, during a chronic infection and uncured response, CD4 T cells don't call upon CD8 T cells. CD8 T cells are not capable of binding to the constantly mutated virus, and this leads to exhausted T cells shown by an increased production of inhibitory receptors such as PD-1. The graph on the bottom right shows CD4 T cells not being able to call upon CD8 T cells, and this leading to a weaker CD8 T cell response and continuous infection. The innate immune response includes inflammation, and evasion of host innate immune response can lead to chronic diseases. Chronic HCV infection causes liver disease through alternating the host lipid metabolism. It increases lipogenesis and impairs both lipid degradation and export, leading to accumulation of lipids inside the host hepatocytes. This manifests as hepatic steatosis, or fatty liver. Accumulation of lipids inside hepatocytes can lead to inflammation of the liver cells, which results in scarring of liver tissue, cirrhosis, and liver failure. So as you can see here, lipid synthesis is increased, and secretion and catabolism is decreased. Hepatocellular carcinoma. Chronic inflammation from both cytokines released during the innate immune response. Hepatic steatosis due to an accumulation of lipids in liver cells and other disruptions in the host hepatocytes can lead to cirrhosis or scarring of the liver tissue. 70% of HCV infections lead to chronic infection. 10 to 20% of people with chronic hepatitis will progress to cirrhosis, and 1 to 5% of people with cirrhosis will progress to hepatocellular carcinoma. And so as you can see here, this is the normal liver, and this is what HCC looks like. Thank you.
Hi everyone, my name is Victoria, and we'll continue with the phylogeny and evolution of hepatitis C virus, or HCV. So HCV is classified into 7 genotypes as shown by the phylogenetic tree on the right. Correlation has been shown between the different genotypes in their response to antiviral treatment and host immune response. There are currently 2 hypotheses for the origin of HCV. The first is zoonotic transmission from other mammals. The second is that HCV infected humans and their ancestors throughout evolutionary history. There has yet been definitive evidence proving either of the 2 hypotheses, and this remains an open debate. There are several factors contributing to the genetic diversity. The first is point mutations due to the error-prone RDRP, and the second is recombination. Currently, there are several treatment options available for HCV infection. The first are antiviral medications or direct-acting antiviral agents, DAAs. There are 7 of the antiviral medications so far, most of them targeting the non-structural proteins 5 and non-structural proteins 3, 4A. In the cases of chronic infection leading to severe liver cirrhosis, liver failure, and liver cancer, patients may require liver transplantation. With the treatment options available, there are still several limitations. The first is that HCV infections are rarely symptomatic before the onset of advanced liver disease, and HCV screening is rare in most parts of the world. The cost of treatments are also high in many countries, so only a subset of individuals are treated, and immunity after effective treatment is insufficient to prevent reinfection. Clinical studies have also shown the development of resistance to antiviral medications. Currently, there are no effective vaccine treatment against hepatitis C virus, and difficulties to develop an effective vaccine includes the extensive genetic diversity due to the error-prone RDRP and the 7 genotypes giving rise to distinct genotype-specific immune responses. In addition, the lack of an adequate animal model also hindered the development for the HCV vaccine. Recent studies have discovered hepativirus as an analog that can be used as an animal model for HCV. However, despite their structural similarity, the 2 of them share relatively low sequence homology. There are several possible vaccine targets. The first is envelope proteins E1 and E2 that mediate entry into hepatocytes with the structure of E2 shown on the right. The second are non-structural proteins NS3, NS4, and NS5, which are dominant targets of CD8 T cells and targets of antiviral medications. In conclusion, HCV is a group 4 plus single-strand RNA virus discovered by Dr. Harvey J., Dr. Michael Houghton, and Dr. Charles Rice, and was named in 1989. Limitations in current diagnosis and treatments for patients and the lack of effective vaccine hinder the eradication of HCV, and thus are the reasons why HCV is still prevalent today. Potential targets of the vaccine are viral glycoproteins E1 and E2, non-structural proteins NS3, 4, and 5. In the future, the World Health Organization set goal to eradicate viral hepatitis as a public health threat by 2030, and the development of an effective vaccine is critical to achieve this goal. In the vaccine development process, there are still a few unanswered questions. The first is, what are the differences in host immune response for each HCV genotype? The second is that what are the mechanisms that allows for antigen-specific immune cell populations to mediate the long-term protection against HCV?
All right, thank you for watching, and that concludes the presentation. Here are the references. Now we open the floor for any questions.